Hello students, a warm welcome to all of you in BioDesire. In today's session, we discuss about the methods and biology. Okay, in methods of biology, we discuss about the different techniques. Like we discuss about the what is microscopic technique, what is the UV visible, what is the spectroscopic technique, what is the PCR, what is the sequencing, etc. Okay, so we start today's session and understand what is the first we discuss about what is the microscope. Okay, what is the microscope? Microscope is a technique which basically is an instrument that are used to visualize the biological specimen. Okay? What is microscope? Microscope is a technique, is a instrument that are used to visualize the biological specimen that not be visualized by the naked eyes. Basically, microscope is a visualizing instrument. Okay? And by the help of this microscope plays a most important role in a research field. Without microscope, we can't perform any single task. Okay, and there are mainly on the basis of visualization. Now, there are two types of microscopic technique. First is your light microscope, and second is your electron microscope. Okay? Light microscope or electron microscope. In light microscope, we visualize the biological specimen due to light. And electron microscope, we visualize the specimen due to electrons. Okay, and the magnification power of electron microscope is more than the light microscope. Light microscope magnify the object approximately 1000 to 1500 times. Okay, in light microscope and in microscope technique, we use a lenses. Okay? This lens to focus the light on a specimen, then magnify or then produce the image. Okay? This microscope is normally this microscope magnify varies gently depending on the type, number of lens, and that make up the microscope. Okay? Depending on the number of microscope, there are two types of microscope. First is your simple light microscope, second is your compound microscope. In simple microscope, we use only one. Okay, only one lens. But in compound microscope, we use two set of lens. First is your objective lens, and second is your eyepiece lens. Okay. Now we discuss about the types of microscope. Okay. Now we discuss about the basic principle of microscope technique. Basic principle of light microscope technique. Okay. The light microscopic technique work on a principle of optical magnification using the visible light. Okay. It uses the lenses to reflect the light ray and magnify the small objects like your cells, tissues, microbes, making them visible to the human eyes. Okay. And now what are the key principles? Key principle is illumination with visible light. Okay. A beam of visible light is directed onto the specimen. Then it will pass through this and reflect First is a illumination with light. A light beam, a visible light beam is directed onto the specimen. Then it will pass through the specimen or reflect through this. Okay. Then reflection by the lenses means the objective lens collect this light from the specimen and form the magnified image. Means this is the objective light. This is the our a specimen which we want to visualize. Then a light is passed through this. Some light is passed through it and some light is reflected. Then the light is passed through this specimen. Okay. When now this light is collected by the objective lens and and the and magnify the image. Okay. Here the eyepiece lens is also present. This eyepiece lens is also magnify the image for the observer, and this is known as a optical magnification. Okay. First, first is your light, and this light is ob- passed through the biological specimen. Okay, and through this specimen, this light is reflect. Okay, and this reflected light is collected by the objective lens. Now this objective lens magnify this image. Okay, then it will light is reach to the eyepiece lens. Now this eyepiece lens again magnify this image, and now a person is easily visualize the object. Okay, and this complete process is known as a optical magnification. First, this optical objective lens magnify the image, then this eyepiece lens magnify the lenses, and this finally eyepiece lens, by the help of this eyepiece lens we easily visualize the biological specimen or your objective. Okay, image formation, this image is real, inverted or magnified image is formed by the objective lens. Then eyepiece lens again enlarge this image and real but enlarge this image. Okay? Objective lens form this image real but inverted and also magnified. Then this inverted or magnified image is further magnified by the eyepiece lens, and then we easily visualize the large image. Okay. Resolution power. What is the resolution power? Resolution power is the ability to distinguish the two close objective as we separated. Okay, and it's depend upon the wavelength of light and numerical aperture of the lens. Okay, this is the basic principle of microscopic technique, and this is the microscope. This is the eyepiece lens. This is your objective lens. This is your space. This is the stage, and this is the illuminator, means light source. Now this light source is passed through this stage. Then it will pass through this specimen. Then it collect by the objective lens. Then eyepiece lens magnify these images. Okay. Now we discuss the key component of light microscope. First, we require the eyepiece lens. This is also known as the ocular lens. This lens is used for the lens you look through. Okay, means this is the lens by which we easily visualize the image, and the magnification power is 10x to 15x. Objective lens, that is located on the rotative nose piece, usually included the 4x, 10x, 40x and 100x for the oil luminescence. Condenser lens means focus the light onto the specimen. Okay, mirror and light sources means reflect or produce the light through the specimen. These are the optical component of your light microscope. Now we discuss about the types of microscope. This microscope techniques is divided into two types. First is your simple microscope. Second is your compound microscope. Now this compound microscope is already also divided into different types like bright field microscope, dark field microscope, fluorescence microscope, confocal microscope, phase contrast microscope, phase contrast microscope, etc. Okay. In bright field microscope, dark field microscope, fluorescence microscope, confocal microscope, and phase contrast microscope. These are the different types of light microscope technique. Simple microscope only contain a single convex lens. Magnification power is 10x to 15x. Compound microscope contain the multiple lens like objective lens as well as eyepiece lens. Magnification power is 1000 to 2000. Okay, and illuminator is a transmitted light. Objective are a thin section of tissues and light, tissues and cells. Okay, now hmm, now this bright field microscope. Okay, in bright field microscope, light directly pass through the pass through the specimen. Okay, in dark field microscope, we use a special condenser. Condenser, it will scatter the light. Okay, it will scatter the light. Then light is reached to our eyes and the magnification power is more than the bright field microscope. Okay, then in phase contrast microscope, here we use a pinhole. On this pinhole, only transmitted light is passed. Okay, if only transmitted light is passed, then the magnification power or the bright and the image of your specimen is better than the dark field microscope or bright field microscope. Okay, in this fluorescence microscope, we take our biological molecule with fluoropores, and whenever this light is passed through this fluoropore, then this fluoropore emits some light and this emitted light is detected by the detector, and the magnification power of this microscope is better than the phase contrast, dark field or bright field. In confocal microscope, here we also use the pinhole, and this pinhole increases the magnification power and increase and now in phase contrast in next is the in phase contrast we use pinhole. Okay, that this pinhole, due to this pinhole, the only transmitted light is passed. Now the image is very clear or clear or easy. The magnification power of now confocal microscope is same as the fluorescence microscope, but here we use the pinhole. On this pinhole only transmitted light is passed and the magnification power of image is very Hi, by the help of confocal microscope, we easily construct the 3D structure of images. 3D structure of your image. Okay, this is the these are the different types of light microscope. Bright microscope, on light is directly passed through the specimen. Dark field microscope, here we use a special type of condenser. Due to this condenser, and due to this condenser, only scattered light is reached to our eyes, and the visualization power and the contrast is very now. hmm, Now these are the different types of microscope: bright, dark, fluorescence, confocal, phase contrast. In bright field, light is directly passed through the specimen. In dark field, light is here. In light field, we use a condenser. In fluorescence microscope, we use a fluorophore molecule. In chlorophyll, in confocal microscope, we use a fluorophore molecule along with the pinhole. In phase contrast, we also use the pinhole system, or we create a and in this phase contrast microscope we also use the pinhole that will enhance the and that enhance the and in phase contrast microscope we use a pinhole system. In this by due to the using this pinhole system, it will enhance the contrast of transparent specimen. Okay, in phase contrast microscope we use a unstained. In by the help of phase contrast microscope, we easily visualize the unstaining specimen. Means whenever we visualize the images by the help of phase contrast microscope, then staining is not required. Whenever we visualize the molecule with bright field microscopy, then staining is required. And in case of fluorophore or confocal microscope, we require a fluorophore molecule, because whenever we take our target molecule with fluorophore molecule, then when the laser light is passed, whenever the laser light it fall onto the fluorophore molecule, then this fluorophore molecule emit the light, and by the help of this light we easily visualize the image. Okay, by the help of confocal microscopic we easily visualize the 3D structure of images. Okay, these are the different types of microscopic technique. Okay, now what is the advantages of microscopic technique? It is relatively easy to use, small and lightweight. It offers the high level of observability and quality. It is unaffected by the electromagnetic rays, do not require the radiation for to operation, require a very little training, allow to observe the living organisms, have the minor maintenance cost compared to the other models, fully adjustable in comfort level for the user. Now disadvantages means the resolution power is very low, make it challenging to view the internal structures, cannot operate in dark field, cannot provide the 3D rendering, and very low magnification caps.